Thank you for that. So a slight change of tactics here, because you've heard a lot of talks about biomarker discovery using various cell lines, etc., and a lot on patient stratification and prognosis. But I'm actually going to present more around the translational gap that you have in early phase clinical trials. So really trying to address the pharmacodynamic issues around biomarkers. So try to address the question, does the drug do what it says it does in a clinical setting? So, I work at Cancer Research UK. So just to start off the talk with some sobering thoughts, actually, at least within the UK, there are over 300,000 new cases of cancer which are diagnosed every year. And, getting cancer is one of the greatest fears that we have across the globe. Someone in the UK is diagnosed with cancer every 2 minutes, and 1 in 2 people in the UK will actually develop cancer at some point in their life. So globally, it is predicted that the incidence of cancer will double over the next 20 years. So these are stats based on the information that we have within Cancer Research UK. So because the disease is because the disease that it actually kills through metastases, we really need to get some more new treatments. Long gone are the days of chemotherapy, and the scientific landscape is changing to having a bit more targeted therapies. So to address this question, what is Cancer Research UK doing to provide some new solutions? So within Cancer Research UK, I work in the Centre for Drug Development. So we're based in London, and what we really do is sponsor early phase clinical trials, mostly phase 1, phase 2. And we have at any given point about 30 projects. They cover a wide range of agents and tumor types. So we focus on the Cancer Research UK priority areas, which are things like pancreatic, lung, brain, and esophageal. In terms of modalities, we have a wide range, ranging from cell therapy, gene therapy, small molecule antibodies, and imaging agents. One of the key attributes that we do bring to the drug development area is a global partnership. So we have a team which actually works with academia, biotechs, and various industries to try and actually develop some of the more emerging novel concepts that, let's say, industry feels a bit too risky to take on, or the biotechs or academia don't have the actual expertise to do that. So we've taken over 100 compounds into early phase clinical trials, and 6 of them have reached the market, including temozolomide and abiraterone. So what is the focus of CDD? So as I mentioned, really we're trying to work on the late preclinical development stage and the early phase clinical trial bit with sourcing projects from academia, biotech, and pharma. I'm just going to present to you some information on the kind of companies that we work with, the drugs, and the kind of activities that we have. So as you can see, we work with companies like Merck, Astex, MedImmune, AZ, BioInvent, Amgen, GSK, and most of these agents are actually first-in-class molecules. So we are really trying to down prioritize the Me Too type approaches and really work on those novel, exciting compounds. So our sweet spot is around translation from preclinical to first-in-human. As I mentioned, first-in-class agents are highly desirable, and we put a real big emphasis on biomarkers. So what we feel is that, really to address phase 2 attrition later down on the line, if we can't demonstrate that the drug is doing what it's supposed to be doing, so addressing the question, have you got the right dose and the right scheduling, then we feel, there is no point in taking the drug forward any further. So I'm going to present to you a couple of case studies, hopefully to give you some examples on how we actually use pharmacodynamic biomarkers in our trials. The first one is actually a Kylob, which is a CD40 targeting antibody. And as we know, CD40 is expressed on B cells amongst other immune cells. And this study briefly was actually a dose escalation study in 28 patients, and you can see the dose ranges that we tested there. And really the primary endpoint and the aim of this study was actually to look at safety tolerability as you would do in a phase 1 trial, but in addition, PK hacker responses, and more importantly, pharmacodynamic responses. The idea behind that is that, if we can achieve a tolerable safety pharmacodynamically driven dose, then we could actually propose that as our recommended phase 2 dose to take forward. So the graph here, what you see is the B cell number with the 50 milligram and with the 150 milligram, and along the x-axis the time, the time of response. So what I hope you can see is that as you increase the dose here, you see a greater knockdown in the majority of the patients in terms of B cells, and the duration actually lasting for up to 48 days with this antibody. So as we increase the dose to going up to 200 mg as well, we see this consistent pattern. So clearly there was no additional benefit seen beyond the 160 mg per dose as we increase the doses. Correspondingly, what we saw was at the lower doses a hacker response that we lost at the high doses. So here we have a molecule that is well tolerated, and we've managed to show a significant decrease in B cell knockdown, particularly, as I mentioned, at the doses above 160. We also looked at some other exploratory and research biomarkers, looking at immune cell numbers, cytokine profiling, as you would do in some of these studies, and really to build a more concrete and a confident picture around what this drug is doing in an early phase study. And we believe that the Haka responses were lost at the higher doses, primarily due to the B cells being depleted. So at the end of this study, we were pretty confident that we've got a drug that is safe, that is well tolerated, can modulate a pharmacodynamic response, and there was no beneficial effect from a biological point of view to shift the dose much higher than the doses that we've tested. The second case study is actually on a SARC inhibitor. So this is sarcatidine, an AZ compound. And briefly, this study, in addition to looking at your safety and tolerability, we were, during the dose escalation, we were looking at two key biomarkers. That's serum CTX and the urinary NTX to creatine ratio. These are induced as inhibition of osteoclast activity, and again, the idea here that we wanted to demonstrate was, have you got the right dose and the right scheduling from this phase 1 study. So these kind of studies, really you don't need that many patients, so we can actually address these things with about 30 patients in a typical phase 1 setting. So what you see here is the serum CTX levels, and as you increase the dose from 50 mg to 125 and 170, I hope I can convince you that you see a marked inhibition of the CTX response, which plateaus out around 28 days. A similar pattern being evident for NTX and creatine. So this kind of data really supports in your first in human, first in class type study that you've engaged your target and you can actually measure a biological response. The drug is doing what it's supposed to be doing. We can select the dose and the scheduling based on such data. The third example I'm going to talk to you about is a Aurora kinase inhibitor. It's Aurora kinase B and C. Specific inhibitor, the GSK compound, and in a phase 1 study, 37 patients in total, but 24 treated at MTD. So in this particular study, we dose escalated up to MTD, but only included the biomarkers in terms of understanding the biology in your expansion arm. So this was done in advanced cancer patients, solid tumors, with a compound being administered 1 hour IV infusion 5 days for every 21 days. So classical phase 1 endpoints, MTD, and looking at PK profile and safety. But we also incorporated a lot of tertiary and exploratory biomarkers, including skin biopsies, tumor biopsies, and circulating markers of cell death like M30 and M65. So with this study, what we did was we looked at the tumors to look at a proof of mechanism marker. This is phosphohistone H3, a well-characterized marker for Aurora kinase activity. And what we saw in the 4 patients treated at the dose expansion in a particular solid tumor setting, 3 patients were showing a decrease following dosing in phosphohistone H3. So we were pretty pleased by seeing the results to show that we've given this drug at a dose just above below MTD, and we're actually picking up modulation of our target. So unfortunately, where this kind of opened up a new set of questions, which I'll come on to in a couple of slides later on. So although we were able to show biological activity as demonstrated by a classic marker, we actually found no evidence of clinical activity. Neither did we find any effects on circulating cell death markers like M30, M65. And neither could we find any PD modulation in a surrogate tissue. So we were hoping we could actually use skin biopsies as a surrogate. So it kind of left us with a bit of questions here, saying, because there was no correlation between the PD modulation and the clinical outcome. So going back to one of the previous presentations, have you got the right patient group here? So, it was a dose expansion study in a set patient population. So, maybe we got the right dose and the right scheduling, but have you got the right patient group? But then we question as well, did we actually get the right dose and the right scheduling, because we actually dose escalate based on an MTD approach rather than a biologically effective dose. And it also opened up more questions around the endpoint that we looked at here was an immunohistochemistry-based endpoint, which, as you all know, is more qualitative rather than quantitative. So do we need to incorporate more quantitative endpoints in making these kind of decisions? I'll come on to some of these challenges that we face with early phase oncology trials later on. So within Center for Drug Development, we kind of have like a toolbox for measuring pharmacodynamic responses. So each study is very unique, and we kind of look into this toolbox and see, what is it that we really want to measure in this study. So typically our phase 1 studies, we would have a dose escalation part, and then once we've figured out a dose that we want to take into, we would have a dose expansion arm. The dose expansion arm would be enriched. So this is different from stratification. So we're not looking for a biomarker positive, biomarker negative arm, but we're looking for a patient group that expresses the target and then try to see whether we can actually modulate a particular response. So a range of tumor markers that we would use routinely in our studies. Some of these things have been talked about today and yesterday. And we do try to incorporate a lot of surrogate markers. So typically, the tumor markers we would obtain only in our dose expansion, because that's where we would get the hard-to-obtain biopsies. And in the surrogate markers, we would typically incorporate them into our dose escalation, because that would be looking at, say, a urine circulating marker or hair follicles. One of the things that we're doing a lot of at the moment is, really expanding the use of ctDNA and CTCs, as well as really branching out in the use of flow cytometry, because there are so many number of assays that we could do in terms of demonstrating biological activity using these technologies. So going back to my question around the challenges that we had with the Aurora kinase study, so the whole problem that we have in terms of the real world around the PD modulation is there's little evidence to correlate PD effect with clinical outcomes. So when you go and speak to a clinician, and they would always put clinical response rightly at the forefront. So if you do a clinical trial and you see a clinical response, forget the PD. You got a good thing there. So let's get going. But if the problem comes when you don't have a clinical response, as in majority of the phase 1 trials that we do, we are struggling to get a PR. If at best. So then what are you left with? You're left with a safety and a tox study and nothing else. And this day and age, that is simply not good enough, and we really want to show something a bit more. This is kind of where your PD response really becomes critical for taking this compound forward when you don't have a clinical response. I think there was some brief mention about this, around how long, what kind of a PD response do we want to see? What is that magnitude of effect? Somebody talked about that early on. How do you actually define what kind of change is a good enough change when you look at biomarkers? We get people from various industry coming to us and say they want to see a 40% change, 50% change, and when we ask how did you get that number, they just do that. So it's one of those things we really don't understand. And what tissue type do you want to see this PD effect? Are you going to be happy with just the tumor, or do you need tumor and surrogate? But then, if you want to obtain this evidence in the tumor, you really have one shot at goal. It's very limited supply that you have in terms of getting repeatable biopsies. So where do you prioritize? So you might want your tumor in a FFPE material for one assay. In the other assay, you might want it fresh frozen. So how can you get all that information with a really small piece of tumor sample, or tumor samples that are difficult to obtain, something like pancreatic? So there is a very little correlation as well between your tumor and your blood-based markers. So these are quite sort of real-life challenges that we face when we're trying to put a data package together in a study report to explain what the drug is doing. And it's also more complicated in the area that we work with, because most of our targets, nearly 90%, are first-in-class agents. So these are novel agents that not many of you have worked on, and you're testing your proof of mechanism assay for the first time. So it's the first time in the clinic with a novel assay, novel target, and you're expecting your biomarker to do a lot. There's a high risk of failure there, and I can talk to you more about those failures when time permits. Well, one of the things that a lot of people don't appreciate is that it really takes a long time to have a good biomarker ready to use in the clinical setting. So I'm talking about using this in a clinical setting in a phase 1 study, and you need to start this in parallel to identifying a new drug, and not when you just identify your lead compound and then you think, okay, let's try and find a biomarker. It takes investment both in time, cost, and resource. And you heard earlier on about the whole issue around resistance and tumor heterogeneity, and this is something very, apparent in this setting, and trying to overcome this is a big challenge. So a couple of take-home messages. So I strongly believe that biomarkers are key to the success of early phase oncology trial, and this is I'm talking about pharmacodynamic biomarkers, and help minimize the phase 2 attrition. So certainly within Center of Drug Development, we really try to make robust decisions based on the end of our phase 1 trials. We are not too keen to pursue a program if it's just only PK, safety, and tox, and we haven't got either a clinical response or a biomarker in there. And I think it's really important to be clear right from the onset, what is it that you want to use from a particular trial design with regard to your marker, and during which part of your study design. So as I said, typically we have an escalation arm and an expansion arm. So expansion arm is often relying on the biopsies, but be realistic about what you want to expect out of that study. So I'm elaborating. It is challenging to discover, develop, and have a robust clinical assay, particularly for a first-in-class agent. But we strongly believe that without having such pharmacodynamic assays, minimizing any phase 2 attrition will remain a significantly higher hurdle. Thank you for that. Just in time. I'm here partly also to seek help from you guys and see if there's anybody who's got any projects or compounds which are in the late stage preclinical development, which are, say, first-in-class agents, or a technology that you would want to apply into a clinical setting. I'm really welcome to hear from you guys, because we really try to work with those folks in academia and in industry or biotech who really got some novel, exciting agents which the charity can support running in a clinical trial. So please come and talk to me. Thank you. Any questions?
Thank you very much, Sid, for this excellent overview and